Otherwise, it might.
All right. Good morning, evening, or good afternoon to some other people. I hope everyone's doing well. This is the very first webinar from Smart MCs, and we are today hosting Professor Steve O in our webinar. And the main focus will be on mesenchymal stem cells and latest approvals, and of course, a lot of cool insights that Steve will be sharing of his research and his insights on the future of the field. So today's webinar is brought to you with Smart MCs. We are a biomaterial company focused on smart microcarriers, hydrogels, and microfluidic solutions. And now I would like to pass on the microphone to Professor Steve O to give first a quick introduction of himself. After that, he can Steve, please.
Yeah, thanks, Fire. So welcome, everyone. Thanks for turning up. For some of you, probably late at night, some early in the morning, to this talk on mesenchymal stem cell therapies or mesenchymal stromal cell therapies, promises fulfilled and potential new applications. So a bit of a background of myself. I was 22 years with the Agency for Science, Technology, and Research. And now I'm currently a scientific advisor to various companies, including Smart MCs and Exterma. And you'll hear some of these research work that we've been doing together. I'm also sitting on the ISCT Asia Regional Executive Committee, which is the premier society for cell and gene therapy. So we try to increase the membership of folks around the Asia-Pac area and upgrade the skills so that we can execute these new therapies. So the outline of the talk will be the following. I'll talk about the various functions of MSCs and their sources, give you some examples of already authorized therapies and their clinical successes, and then talk a little bit about how machine learning can be applied for one example of osteoarthritis treatment, some outline of the future versions of MSCs that are coming into the space, and how new modalities like biodegradable microcarriers and non-DMSO cryopreservations will improve the therapies in the next few years. And finally, importantly, also to share with you some of these ICT membership and the benefits of joining the society. So many of you probably know of the mesenchymal stromal cells as the cell types that can differentiate to muscles, osteoblasts, chondrocytes, and adipocytes. That was kind of the standard criteria for determining MSCs. And in some cases, they could trans-differentiate to the endoderm lineage and the ectoderm lineage. More recently, however, probably in the last 5 years or so, we've discovered that these cells are pretty versatile. They have a whole bunch of ability to secrete paracrines, immunomodulatory activities. They can integrate into the tissue. Besides the soluble paracrines, they also secrete these extracellular vesicles, which are nanoparticle-sized structures. They could even transfer large mitochondria between cells from them to other cells, and they can mediate immune cells activity through cell-cell contact. On top of that, they have antimicrobial capabilities to secrete antimicrobial peptides. There's a whole bunch of angiogenic factors that are secreted. They can, some of these can regenerate the tissues, some of these factors. Also discovered to have the ability to kill tumors. And they have antioxidative secretions, as well as anti-apoptotic capabilities. They can downregulate the activities of macrophages and T cells. So a whole bunch of activities, and these cells come from a wide variety of sources, such as bone marrow, placenta, adipose, Wharton jellies, dental pulp, and endometrium, dermis, and even breast milk. What I've circled here are the commonly used sources of MSCs, and they are the ones that are both autologous and allogeneic. So the younger ones like the placenta and the Wharton jelly cell sources would be able to give you younger MSCs to expand out to multiple population doubling so that you can provide the allogeneic therapies. However, the bioprocess that ensues after isolation is important to think about as well. So you have your donor status, which impacts the quality of the starting materials. I talked about the tissue of origin. So the younger sources would also impact on the proliferation rate. The method of isolation would determine how much cells that you can isolate from the source. For example, a biopsy followed by enzymatic, and then maybe some sort of sorting would lead to reduced numbers of starting cell population. Then when you go into the production, you have to deal with the media optimization, oxygen levels, percentage confluences, surface areas, and a whole bunch of other factors, whether they're in flask or bioreactors. And finally, you also have to preserve the cells. Right now, there are not that many choices out there besides DMSO. Then you have to rescue them before you put them back into the patient. So my background being in process development, we have to deal with a whole bunch of activities in the process scale up, optimizing the culturing conditions to provide a preclinical supply, and then finally packaging and delivery. So some of these concepts and trainings are actually provided by the Workforce Development and Biomanufacturing as part of the global partnership between ISCT and CMAT. And you can learn about these concepts, production and supply models, critical quality attributes, risk case development of the process, and so forth. So some of the inputs would be things like the media, the growth factor, and the microcarriers, since MSCs are anchorage dependent, oxygen, pH, even agitation and aeration methods will determine whether the cells grow as a monolayer or clump. Different seeding densities will lead to higher or lower cell use. The limitation of passage number, how to harvest efficiently, and the frequency of feeding, which will then affect all of these, your cell yields, your viability, perhaps some of the phenotypic markers, potency, and the functional assays. So these are the kind of things you have to work on, process changes, troubleshooting, quality by design, in order to get high quality outputs from these inputs. And then you have to go into a documentation of these methods so that you have a GMP quality process to improve the product and to ensure consistent, safe, pure, and potent products. These are some of the issues that companies, which I'll mention later, have to deal with in order to satisfy the regulators on a high quality product. So finally, when you've got the product, you have to look at injection methods, whether it's a local or an IV injection, the injection sites, the type of device used, the buffers, and whether you have any carriers, for example, in the target tissue. And then the recipients, depending on the stage of the disease severity, there would be different responses to it. Also, whether the tissue is hypoxic or there's stiffness in the extracellular matrix and so forth. It will finally determine the outcome of MSCs. So now I'll move on to giving you some examples of MSC therapies that have already been shown to be successful. Here you see a patient with severe acute skin graft versus host disease infused with these MSCs. And 3 weeks later, you can see this response of the patient has cleared up the skin lesions significantly. Another example is regeneration of the cartilage or reduced inflammation. So where MSCs are injected directly to the defective cartilage, you can see some restoration of the cartilage lesions here. A third example is in the critical limb ischemia, where there's poor angiogenesis around the lesion where there is the ulcer. So by injecting at multiple points around the ulcer, you can get a restoration of healing of the ulcer. And in this case, it's a partial restoration of healing of the ulcer. So all these examples are examples where therapies have been authorized in different parts of the world. The final example is in reducing major adverse cardiovascular events in chronic heart failure. So this was a study of a bunch of studies in the papers showing that injection of MSCs from different sources can reduce mortality rate, improve the left ventricular function, and also reduce the scar size. Now, to date, since around 2010, when the first therapies were authorized, was in connective tissue disorders in Korea, then acute myocardial infarction, and Crohn's fistula. So Korea was kind of in the leadership area for MSC approvals, and CardiStem also for osteoarthritis. Then the Western countries started to get authorization for graft versus host disease, Canada and Australia, New Zealand. And then followed by Japan for GVHD again. And India had their first one with critical limb ischemia by Stempeutics. And then an international one with Takeda and Tygenix for Crohn's fistula. Iran had one for osteoarthritis, and then Japan again for spinal cord injury. But the biggest market, US and China, in quick succession started to authorize BM and umbilical cord blood-derived MSCs for steroid refractory acute graft versus host disease. So this is a big game changer, because these are the two biggest markets which had helped back from authorizing MSCs for therapies. So we can expect in the 2025 that there's going to be more of these therapies coming into play with the cells, for example, in heart failure. So given the track record of Mesoblast has been longest in this space, they submitted their first the first submission was done in 2020, but the FDA came back to say, oh, we needed to look at more data from the adults or another a set of data from children for evidence of the effectiveness in severe refractory graft versus host disease. So 3 years later, when they submitted it, showing lots of safety data and functionality and potency, they still didn't get authorization. But with persistence, in December 2024, finally they got the approval, and I think the whole field was celebrating that finally the US FDA saw that MSCs is really a therapy that can make a difference in patients. So the impact of graft versus host disease, around 10,000 bone marrow transplants a year, and 1,500 children are affected. Half of them develop acute GVHD and are resistant to standard treatments. So this drug is life-saving for children and their families, says Joanne Hertzberg, who's one of the leading authorities in this space. And the Mesoblast is planning to launch Ryoncil at a price similar to other cell and gene therapies. and they plan to expand it to adults and other inflammatory conditions. And in 2025, there will be another product called Revascor for heart failure in the future. So in terms of pricing, when the first products came out, they were in the 5,000 range for treatment, to CardiStem for cartilage repair for 20,000, TEMCELL, GVHD for 170,000, Prochymal. So this is likely to be the price point for the newly approved therapies in the US as well. Okay, so I'm going to spend a little bit of time on the importance of machine learning, as this is the foundation of many of the AI research that's ongoing. This work was done about 3 years ago, and we've seen some good recommendations coming out of applying machine learning to literature research. So what we did here was that we looked at the the treatment cases in blue, and the actual treatments are in blue, and red is the machine learning simulating every one of those cases 1 million times for the best treatment for effective repair. So MP stands for maximum pain repair, maximum pain, sorry. MW is maximum WOMAC. LP is long-term pain, and LW is long-term WOMAC. So importantly, an effective score is anything above 60%, which is shown by the black line. And with the machine learning, we showed that it's possible to actually get much better care, repair with the red line above the 60% in every case, except for perhaps long-term WOMAC wasn't as good as everything else. But most of these effective repair or pain, WOMAC, long-term pain and long-term WOMAC was much better than the actual literature based on a method that we had developed to come up with the cell density and dose. So this is the simulation. We found that for maximum pain, the cell dose CC was in the range of 4 to 7.6 million. For long-term pain, and then the dose was 1 to 29 million. For long-term pain, they range from 6.7 to 15.1 and 1 to 198 million cells total. Whereas for maximum WOMAC, which is a measure of osteoarthritic recovery, the parameters were different. The range was still the same, but the dose was 41 to 198 and 41 to 67. Of course, these are just simulations. What we'd like to do in the years going forward is to actually try these doses to see if they work as well. But this slide kind of summarizes it. If a patient comes in with a certain diagnosis and we want to have maximum pain, the treatment recommendation might be, say, medium dose to high cell concentration. For long-term pain, it may be medium dose and medium cell concentration. Whereas for maximum cartilage repair, it could be high dose, high cell concentration, and so forth. So so rather than giving just one concentration and one dose, machine learning will tell us how to better treat patients to maximize their recovery, whether it's pain or a WOMAC score, which is a measure of cartilage repair. So we get an opportunity to do this in Singapore. The Singapore Ministry of Health has allowed the conditional use of autologous MSCs for cartilage repair under constrained use framework for a period of 36 months. And then the healthcare institutions or the companies that I'm aiding, like StemCourt, would be able to submit the data. So we will look at applying some of these suggestions by machine learning at different cell concentrations and doses to see if we can get better repair. So moving on to the next stage of the different versions of MSCs, we can see that Cynata, which is another Australian company, has come up with a different way of making MSCs from iPSCs. So they go through expanding the stock iPSCs, then induce them towards the mesoderm, then form a colony, and then finally differentiate to the final MSC from the precursor. So the advantage is that rather than harvest multiple MSC donors and then expand them, they would need multiple infusions per patient. They would have one source essentially that can make a lot of these precursor cells, bank them down, and then also expand them and make lots of banks of MSCs. So in terms of the conventional process, they need a continuous supply of donors, and the population doublings are limited. So whereas with chimeras, there will be one donor, effectively is limitless. So they only need, because of the high replication, they only need 10 population doublings to get the dose, and the infusions is going to be much lower than the bone marrow derived cells. The second new development is in engineering the MSCs themselves. So you can either engineer them with nanoparticles or engineer them to secrete microRNA or non-coding RNA into the exosomes, or genetically engineer the MSCs with an exogene like a plasmid. And then these cells could home to a tumor and kill the tumor. So one such company that's working in Singapore is called AGEMS, and they've targeted the development of MSCs for a brain tumor. So they express thymidine kinase as a plasmid inside the MSCs. So the MSCs are modified. And then they produce enough of the MSCs and then dose them into the tumor. So the phase 1 clinical trial that's being planned is to first resect the major bulk of the tumor, and then inject in these engineered MSCs, sealed it with a fibrin glue, and then they take oral 5-fluorouracil, which will be able to turn on, be activated by the thymidine kinase in the region, and then will kill off the tumor. So that's planned for 2025. So these are the kind of two new approaches to us using MSCs as an examples that's coming. Finally, in terms of the production of the MSCs, working with Smart MCs, we're coming up with new, or we have already new biodegradable microcarriers, which are much better than what's in currently available in the market. So these are the kind of tensile strength that we have found in the literature. Most of the tissues are actually very kind of soft, starting with the brain, the softest, then these endodermal tissues, and then nerves, and finally the bones are the hardest. So typically polystyrene microcarriers tend to be in the very high gigapascal range, but we're able to make microcarriers in the kilopascal range, which are more conducive and similar to the tissue tensile strength of the body. And we think that this is one of the features that is going to be beneficial for the field in terms of providing the right physiology for expansion of the cells. So we've demonstrated this by inoculating the cells on these microcarriers, the X series, and you can see a high attachment rate, about 80% after 6 hours, and exponential growth over 5 days. And we're equivalent, if not slightly better than the commonly used Cytodex-1 in the literature for expansion. So you can see there, these are stained cells covering the microcarriers. For fibroblasts, the attachment time is within 1 to 2 hours, also exponential growth over 8 days. And here the expansion is 3 to 4 times higher than Solihull or Cytodex. So again, see nice confluent layers of microcarriers. Then for virus production, again, the host cells have very fast attachment rate, exponential growth in 3 days, and expansion fold is also double that of the competitive microcarriers. So these are the virus cells covering the microcarriers. And importantly, because we made them out of material that can be dissolved with EEE or small molecule, the cells are completely released. So there's about 90% harvest of the cells from the microcarriers. Whereas from things like Solihull, Cytodex, you have to filter out the hard microcarriers. So sometimes the cells are trapped. So there's about 60% harvest. So you can see here, starting carriers, then fully dissolve, and then starting carriers, cells are released, but then when you filter out the microcarriers, some cells get trapped in the filters. So the second new technology is this non-DMSO cryopreservation solution. So cryopreservation, using DMSO has been found to be a big problem. For example, many difficult to freeze cell types like beta cell, islets, neurons, and iPSCs, the DMSO doesn't work so well. Even T cells behave erratically post-store. And there's problems with clumping, and the potency of the product drops, and they need to optimize the process. So Exterma has developed this solution whereby they can use one molecule that can work as hypothermic storage at both 4 degrees and also be frozen down between -80 to 196 for cryopreservation after manufacturing, and then transport to the patients afterwards. Likewise, the same material can be used to create working cell bank and master cell banks for allogeneic therapies. So DMSO, as mentioned earlier, toxic. There's sometimes viability can be as low as 15%, functionality is impaired. There's also the problem of site injection causing pain and swelling. And also some patients react violently to the injection of DMSO. The cells can also have chromosomal damage and adverse mutations, whereas the FBS itself is batch-to-batch variation and process unfriendly. So the solution is an antifreeze protein. So the antifreeze protein mimetic, which is what the molecule is.
I think Steve might have been disconnected, so apologies everyone. We'll just give it a few seconds.
Is able.
Steve,
Yeah.
disconnected. Sorry.
I've disconnected.
Yeah, now we can hear you.
Okay. How long was I
over,
disconnected?
No, just 30 seconds. So if you can go back to the previous slide and start again.
Yeah. So what I was saying was that the XT Vivo causes the water molecules to freeze in very small crystal, and therefore they can thaw easily, versus water and the antifreeze protein from the on the Arctic fish. And it's chemically defined, doesn't have serum or protein. It's a plug-and-play solution, and there's a GMP-grade solution with a drug master file. So we tested this about 3-4 years ago for MSCs to see the ability to freeze and thaw and compare it to CS10, the 10% DMSO. So part of the issues is that you need to hold the DMSO for a while before you can freeze. So we want to see how long you can hold it and whether the cells will be viable. And also most of the media is moving towards a defined serum-free media. So we have to test whether serum-free media could work in these experiments. So we work with patient-derived MSCs, triplicates. I use the Corning cool cell for freezing, held it for a week, and then we thawed them in a controlled thawing process, and then recovered the cells to grow both in monolayers and microcarriers. So by holding the solution in both serum and serum-free, we saw that for 24 hours, the cells retain their viability. Whereas with the CS10, you can see that by around 4 hours, which is actually the time limit that CS10 says you shouldn't hold it for more than 4 hours, it drops to about 60% after 24 hours. Likewise, in the serum-free condition, it stays high for 4 hours, but then by the next day, there's a significant drop in cell viability. Then after freezing for a week and then thawing, when they were thawed back out, you can see the recovery of the viability in serum was 80%, in CS10 versus 90%. And the cells continue to drop in viability to 60%. Likewise, in serum-free conditions, this is even worse. It drops to about 75% upon thaw, and then stays as low as 60%. And importantly, when we put the cells back to culture, this is the growth of the CS10 cells on microcarriers. It's a very long 4-day lag before the cells grew, whereas cells took off immediately when they were recovered in the serum-free media post-thaw from XD Thrive. So this shows that then the cells the XD Thrive can be used to create a master cell bank expansion to create a working cell bank and make the final product. We also tested that they were able to form various tissue types, the cartilage, bone, and a lipid, and there was no tumorigenesis and no karyotypic mutation. Whereas the CS10 showed one chromosome 10 long arm loss at passage 8. So the conclusion was that there's significantly improved viability before and post-thaw, especially in serum-free media. And this is an important advantage in manufacturing, both for filling and transportation. So the last issue is the injection site issue. So when you inject CS10, you can see this swelling on the pores of animals versus the XD Thrive, there's no such swelling. And this can last for 30 minutes before it returns to normal. So but an additional benefit is you don't get this injection site reaction to patients if you're giving multiple infusions of the MSCs in therapies. So that kind of finishes the technology part of the presentation. I'd like to now spend a few minutes to encourage folks who are not yet members of ISCT to consider membership. So ISCT is a global translational focused expert community. And we want to improve lives through safe and effective cell and gene therapies, as well as drive clinical translation of these therapies worldwide. So this is the only society I know that goes from basic research to process development all the way to clinical translation, and is also advice on commercialization. So this is essentially what I just said. It goes from innovation, translation to clinic, first in man, launch, and product access. So for the last 31 years, most of the growth has been in North America and Europe, but we are now seeing Asia coming up very fast. We've just overtaken Australia and New Zealand, 14%. And soon, I think in the next year or so, we'll be at the same numbers of members as Europe. Because we still have a very big, several countries, China, India, Indonesia, who are not members. So there's both the individual membership. You can also register your more junior staff as a lab membership, and there's also industry membership, where you, if you pay the fees, you can contribute to the shaping of the society. Some example is the Institute of Training and Development. So some courses include things like product handling and regulatory 101, essentials of cell therapy product manufacturing qualification and validation, cell therapy manufacturing optimization and strategy, and then the principles and application of cell therapy manufacturing. So lots of both theory and in some parts of the world, we also provide hands-on courses. We're trying to bring more of that into Asia in the next few years. And then there's lots of open access webinars as a member. So you can cover things, T-cell therapies, cryopreservation, natural killer cell therapies, manufacturing challenges, and so forth. So 2025, we're at New Orleans, May 7th to 10th. It's a bit hard to get to, but it's worth considering attending, because I'm sure there's going to be a lot of sessions on MSCs, EVs, engineered MSCs, and so forth. As Bruce Levine, one of the pioneers in T-cell therapies. Yeah, so please seriously consider joining IC today. Bruce says that it was the society that was foundational in developing his career from a junior researcher all the way to a leader in the T-cell space. So in conclusion, take-home message is MSC therapies are at the cusp of exponential growth after many years. Consider these new microcarrier cryopreservation technologies that will drive manufacturing progress, and join ISCT to upgrade your skills in the cell and gene therapy space. So if there's any further questions or you'd like to reach out, you can reach out to me by this email below, and I'm also available to be connected on LinkedIn. So happy to take questions now. Thank you.
Thank you, Steve, for the great presentation. I think that was a very insightful talk, especially covering, I guess, a big spectrum of things, from all the way from basics of MSCs, how MSCs essentially work, all the way to more detailed stuff on the FDA and cryopreservation and large-scale manufacturing. So for the audience, we're just gonna do a quick question and answers essentially for the next 5 to 10 minutes, and that will be followed also by a bit of career talk with Steve. So please stay tuned. Just wanted to start, I guess, Steve first. What do you think took so long for the MSC field to get some approvals in the regulatory side? So I mean, for example, Mesoblast you mentioned on the FDA, the recent FDA approvals. I think they were founded roughly in 2006. So what do you think took them so long to get here? What was the main challenges?
Yeah, I think there's several aspects to that answer. Many of the therapies, as you saw, they are not life-saving therapies, right? So I think especially in the US, therapies that tend to be authorized when you can prevent cancer that causes death, or heart failure, that causes death, something significant. So things like the repair of the cartilage, or immune modulation, or the Crohn's disease, or the wound healing, they're not transformative therapy. So I think one aspect was that they were not seen as being a significant change in medical practice. And then also because MSCs could do multiple things, right? The authorities wanted to understand mechanism of action and show what is the marker of the potency of the cells. So people had to come up with ways to measure what they're preparing is going to be potent when they input it to the particular therapy. But in those life-saving therapies like T cells, they were not asked to do that. In fact, I heard that one of the markers, which is interferon secretion, which one of the killing mechanisms of the T cells for CAR therapy, that does not correlate with the in vivo performance, but at least they had that as a marker. So, but because the in vivo response was curative over time with multiple patients, they got accelerated approval. But thankfully, this change in the environment by the FDA accelerating all cell and gene therapies is now going to help MSCs turn the corner and be recognized as a therapy that is curative in some cases or regenerative in others.
Yeah, very cool. Yeah, I guess the fact that a lot of the diseases are not so I guess deadly might cause further delay in getting approvals is a big thing. So what were the biggest health risks in these treatments, do you think, when you're being treated with MSCs?
Health risks. I think the safety profiles have always been good. They have to go through the phase 1 safety, right? So MSCs have never had any adverse events in terms of the tumors or most of the time people are afraid that the stem cells becomes a tumor. So in no case has injection into any of these sites cause tumor growth. And in fact, most of the times the cells are cleared in a couple of weeks at the most. So sometimes they're there for 3 weeks, 4 weeks. So their effects is transient. They do their regenerative or immune modulatory activity, then they're gone. So in terms of safety, they're very safe. There's no issues with safety as far as we can see. However, maybe some of the newer engineered cells, they have to demonstrate safety because you're changing the properties of the cells.
Yeah, of course. I can imagine the challenges with modifying the cells and how they can raise a lot of different range of potential risks. That's very cool. So I got a question also on the cryopreservation side from Lynn. So how much impact do you think the crystallization of water inside the cells has compared to the outside of the cells? So I think you were speaking about like the technique on cryopreservation. So I think that's a very big part of I guess if this MSC is gonna come to clinics, cryopreservation will be a big part. So this question I think is referring to that.
Yeah. Yeah, the crystals that are formed inside the cells are the most critical, as they can damage organelles, DNA, and then even cause the cells to lyse if they're very large. So both the process of freezing, creating the crystals, and then thawing can lead to cell destruction or damage to the internal organelles, mitochondria of the cells, which is why you have to get as small a crystalline structure as possible in the in the micron scale, 10 microns or less. Yeah, important to keep that small inside the cell, not so much outside.
Right. That's very cool. And then what do you think on the kind of I guess the new generation of the cell therapies on the MSC part, especially exosomes and extracellular vesicles? So how much do you think they will play a role in the future? Are they going to replace MSCs like as a cell treatment, or do you think they will be kind of further along because of the approvals and how long they might take in terms of getting actually like regulatory approvals?
Yeah, I think because the EVs are made by the cells, right? The challenge will be to again making large doses of the EVs. So I, you probably have to start with those early MSC sources or iPS-derived MSC sources so that you can generate a large uh, numbers of cells, grow out to high passages in order to make the EVs. So the either you have to push up the EVs per cell production, or you have to make a lot of MSCs and then harvest the EVs. And for example, I think that the EVs needs to be in the 10 to the 10 to 10 to the 12 particles per mil dose to be effective. Then when you inject it, if it's IV, is going to get diluted all over your body. So probably the first indications would have to be localized injections for a particular disease where you don't have that wide distribution. So as MSCs have taken us 30 plus years to get to
Mhm.
therapy, I don't think EVs will take any shorter unless they find this
Right.
very niche indication where just a small dose of EVs and you've engineered the EVs to be extremely potent compared to the cells, and then it's transformative. Then you could that could be your first indication where just a limited dose of EVs can change the disease state.
And in terms of your insights, do you think there are less potential risks with EVs in general, or is it like you think it's gonna just it's unknown?
Yeah, I think risk wise, like it's MSCs are already not very risky, so I don't think the EVs secreted are going to be any more risky.
Mhm.
The challenge will be the efficacy and then the targeting, because the EVs don't you don't know how they reach the target tissues in order to be absorbed by the cells. So there probably also needs to be some engineering so that the cells, the EVs are targeting that cell type or tissue type for healing. Yeah, it won't be a safety issue. It'll be delivery issue and dilution issue and production issues. All the things that we had to deal with the MSCs themselves.
All right, that's very cool. I guess that concludes kind of my questions and most of the questions I had in mind and the team. So if anybody has any other question, please drop it in the chat or in the kind of the Q&A. In the meantime, just for the last part of the presentation, Steve, I just wanted to quickly cover a little bit of like your career pathways, how you got where you are now, and understand a bit more for more younger people on how they navigate how can they can navigate essentially through their scientific endeavor. So yeah, if you can just give us a quick I know it's definitely a lot of very fruitful and have been a very hardworking pathway, but if you could summarize like how you got where you are here now and a little bit of talks around the main challenges that you faced throughout the pathway, that would be great.
Okay, thanks, Maya. Yeah, I think my advice to researchers is to find the areas that are most uh, interesting to you and then follow it as deeply as possible. So when I when I started in the bioprocessing field, I started in antibodies. And so I was following at that time those companies in antibody manufacturing, folks like Genentech, Amgen, Roche, and Chiron, companies like this. And then looking at what were the therapies they were developing and what were the challenges in making those cells. Because I was a bioprocess engineer, I looked at things related to manufacturing. The cell line engineering was the one. The second was uh, production in bioreactors, and then purification technology and analytics, and then the clinical trials. So I followed all of those things related to biomanufacturing. Then around 2001, when I switched to stem cells, essentially all that I learned from the biologics, I was kind of revisiting, but in the stem cell space. So what I learned was that even though I had that background, handling cells was so different from handling proteins and biologics. And there were new things that you had to understand, like you had to switch from a suspension culture to work with microcarriers. And then some of the microcarriers that were available didn't do the job. So we had to look at testing a bunch of microcarriers. And then that's what so many years later led to the foundation of Smart MCs in creating these bespoke microcarriers. The other aspect is also keep up to trend with industry news. So I subscribe when I started to 1 or 2 newsletters of industry news, but now it's grown, exponentially. So I would suggest you get subscriptions to things like Bioprocess International or Cell and Gene Therapies, or ISCT also gives regular mailers out, and cytotherapy. So you're keeping abreast of the things that you're interested in. In my case, it's mostly cell therapies, but also keep an eye on the gene therapies, because sometimes the gene therapies can impact the cell therapies either positively or negatively. And then I stick to 2 conferences that are very important. So ICT is one for the industry translation. Then ISSCR is the second one for the stem cell basic biology. And then in between, I will be attending or get invited to other conferences. But both of those societies, I've been with them for 25 years or more. Yeah.
That's very cool. Yeah, no, that's really, really great. And what do you think kind of has been what is different today compared to I guess 10 or 20 years ago, do you think, in academia and also industry in biotech particularly? So do you see it much different, or younger generations should take different pathways compared to back then, or what do you think of that?
Yeah, so I'm starting to use AI a lot, the Chat GPT and Ask AI and stuff. So I think this generation, because there's so much stuff coming at you, you should use these technologies to keep up to date. So now this year I've started to talk to Ask AI regularly on questions. I might even review a paper using ChatGPT Plus, right? So you sent me a paper on the biological corona that sits around the EVs. So I send that to ChatGPT to tell me the critical points of this paper and what are the practical uses of this, and then I'll read the paper and give me a summary. So I would use tools like this to just keep up to date. Of course, I'll still scan that paper, make sure that the review was accurate. Yeah, so that's what I would do. And then now and again, I would like review an area, let's say age reversal, right? I would say, okay, tell me what are the latest technologies in age reversals and what are the genes that are important in age reversal. Then I'll come up with a bunch of answers. So definitely very different from 30 plus years ago, where you have to go to the library.
Yeah, I can imagine how life would have been different without ChatGPT and all the even from Google have been really, really helpful. So do you think in general now is because there's always like thoughts that now the field of, like, for example, biotech is much more populated and there's a lot more work being done, so there's a lot more competition. Do you see that a problem in the academic pathways for people who want to endeavor like getting a postdoc position or getting permanent teaching positions or research positions further down the track? What is your kind of uptake on that compared to what it was 10 years ago, 20 years ago?
Yeah, I think that because of this proliferation of discoveries and technology, there needs to be more cross collaborations of different disciplines, and there probably needs to be bigger teams working together to solve problems that an individual can't solve alone from an early stage. So I would, yeah, I would, unless you've discovered a completely new area of research and you have the first mover advantage. Many of these issues in manufacturing anyway, they require you working with analytics technology, cryopreservation technologies, genetic engineering, so on. So you need to be conversant with other people's disciplines. Biomaterials are also another area. So I think talk to folks that are outside of your main discipline.
So kind of communication, teamwork, and AI. Use
Yes.
those three
Yeah.
tools.
Yeah.
That's very cool. All right. So if there's no other questions, then I guess we're coming to the hour. So thank you very much, Steve, for the great presentation and all the time it took for this talk. Just for the audience, we're gonna be recording this and it will be uploaded on YouTube as we have posted on the website. So you won't be really missing out anything if you have missed parts of this talk. So again, thanks everyone for making it. It's really great to host you, and I hope in the next month's webinars we can have other also kind of scientific discussions here as well. If there's anything else, Steve, you would like to mention before we hop off?
No, thanks for inviting me as the first speaker, and look forward to other talks hosted by Smart MCs. Thank you very much, Brian and the team, for doing this.
Sure. Thank you very much, and it's a pleasure for us. All right.
Cheers.
Thank you
All
very
right.
much.
Good night. Have a good rest.
Bye.